This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 105, recorded on May 28th, 2015. Hey, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Hi there. How you doing?
I'm well. And you?
Fine.
You're getting ready to travel.
Yes, I'm going to the ASM meeting in New Orleans.
Is that something you look forward to?
I certainly do. I see my friends, and I may even get some science in.
Great.
Who knows?
Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello.
You're also traveling, right?
Yes, and I'm looking forward to seeing my friend Elio at New Orleans.
Oh, nice. And also joining us today from New Orleans, Michael Schmidt.
Hello, everyone. I traveled early.
Wow. Is it really hot there?
No, it's not too bad. It's about 85 degrees. The sun is out, and it looks like we're going to have the classic 4 p.m. thunder shower. At least that's what the forecaster said.
Well, 3 out of the 4 of the TWiM team will be there. I will not. I will be here.
That's a shame. That's a shame.
You'll be missed.
Next year, I will go. It's in Boston, and I can't wait to see the combined GM-ICAC.
I'm looking forward to that. I
just
think
for our general listeners, that's the combination of the basic scientists and the clinical microbiologists.
That's right. It should be interesting. You going to go to that one, Michele? Yeah. You have to go.
I'll be there.
Hey, we'll all be there. Very good. Maybe we can do a TWiM right there.
Hey, that'd be neat.
A live TWiM.
Yeah, last year I did the ICAC Live series, which was very well received by. We had standing room only audience members for many of the sessions. So it was.
Of course, Michael, it's you. You're a star. They come to see you.
We have ASM Live this year here at the general meeting with our good friend Stan Malloy.
Yep. Yeah, those are cool. Well, have fun, everyone. And meanwhile, today we will have a podcast for you. And we're going to start off with a snippet from Michael.
Well, I selected this paper because my neighborhood, we're seeing the sprouting of farmers markets everywhere. Charleston is a great place. We have a farmers market on Saturday down in the peninsula, and they're literally everywhere out on the islands. And my university actually has a farmers market every Friday in the Horseshoe. And the local farmers bring in everything from zucchini to tomatoes to watermelons, and it's really pretty neat. And they bring in a whole bunch of fresh herbs that people can select. And the paper I picked for today is one from the United Kingdom, and it's entitled An Assessment of the Microbiological Safety of Fresh Whole Leaf Herbs from Retail Premises in the United Kingdom, with a focus on Salmonella species. Now, Salmonella is important because anytime it's associated with food, it's bad, because Salmonella is always considered a pathogen that's very infectious at a very low dose. So if you find Salmonella associated with food, and we've seen Salmonella in everything from peanut butter to chickens, and anyone who prepares food at home or went through a restaurant training course know how important Salmonella is to the food industry. And so these authors, Carolyn Willis, Lorraine Sandler-Reeves, Nicola Elvis, Heather Arid, Andrew Fox, Moira Kay, Elizabeth DePina, Chris Lane, and Jim McLaughlin, all from the Public Health England Food, Water, and Environmental Microbiology labs that are throughout the United Kingdom. And there was a large cadre of scientists that were involved in the study. At one point in time, 120 public health workers went out And from a period of January to March in 2014, their task was to go out and collect herbs. And they collected everything from basil to bay leaves to something I never heard about, which was Walleria. And
I tried to look it up. Not much on the internet on Walleria.
Yeah, and the interesting thing is, as anyone who's ever been to England know, that the expats who were stationed in India brought back, of course, Indian cuisine to the United Kingdom, and curry is very popular, and so they use a lot of fresh curry leaves and pan leaves. And
wait a minute, I found out that curry leaves are not the same as curry powder.
Really?
Yeah, curry powder is what you put in curry. Curry leaves you add to it in addition to some dishes.
Okay.
It's not quite the same.
Well,
I found out a lot of gastronomic facts here.
I thought, and this is a really fun, well-done paper. And the genesis of this is in 2007, there were 32 cases of Salmonella Sefentenberg infections that were identified in England and Wales, and then there were 19 further ones identified in Scotland, Denmark, the Netherlands, and of course the USA is not exempt from this. And they did all the traditional molecular biology in this, but what they were tasked to do is to now do a survey in 2014 and ask the question, how safe are fresh herbs in the United Kingdom? So they went out to local markets and bought fresh herbs. And then they did a very bread and butter type of microbiological experiment where they took the herbs and they took about 25 grams of herb and they put it into something called a stomacher along with 225 milliliters of appropriate diluent that would effectively macerate the herb to release any of the microbes. And what a stomacher is, if you haven't seen it, is it's as its name implies what our stomach does, where it grinds up our food. Everyone's had the digestive process where we felt the food moving in our stomach, and that's effectively what a stomacher does. It's a really cool device that we have in the lab, and it has paddles, and you put the stuff in, and it macerates up the herbs. And they simply asked a question, were E. coli or Salmonella associated with the herbs? And unfortunately, of the 774 samples that they processed from these 120 different sites, they found quite a bit of contamination. Disturbingly so, that really caused them to sit back and go, what could be going on? And as the story unfolds in the paper, what we learn is our food supply is truly global. The herbs came from both the European community as well as every continent except Antarctica. And of course, Antarctica probably doesn't have a very big farming community. But so it was 6 of the 7 continents where these herbs were being imported to the United Kingdom, an island nation. And
hold on a second, Michael. One thing that struck me is how relatively few samples.
I agree.
Salmonella in it. It's really a study. It's a noble study, but I would have concluded that the food supply is reasonably safe because only 9 out of 774 samples had a significant amount of Salmonella. I consider
without washing them.
Without washing them.
Without washing them.
I wouldn't be so, upset about this result because it seems to me that at least what they looked at is not very, very commonly contaminated with Salmonella or E. coli.
Well, 13% of the samples and 11% of the samples actually had E. coli levels that were higher.
But E. coli, okay, sure.
E. coli, you need a higher dose in order to become ill. And I think that's the take-home message of this paper, is that they concluded that there were 1.2% of the samples had this frank contamination with Salmonella, and 13% had E. coli contamination. But what struck me as being interesting in this paper is the global nature of our food supply and the fact as Elio just pointed out, is it's relatively safe. But they also, in their discussion, go into how many of these fresh herbs are processed. We've all either grown parsley in our kitchen or some other herb on our flower box, and you always have to wash the dirt off. And so consequently, in the commercial world, they do indeed wash these things through commercial tanks. And I think what they're commenting on is the importance of making certain that as the food or herbs are processed, that they're certain that you're using potable water that isn't contaminated, because that's how they think. And that goes back, and they have a neat table showing the geographic location of where the hot spots are in terms of salmonella that's coming in, and what's the location of the things and the percentage of the samples that were contaminated. So it's an interesting approach to looking at how safe is our food supply. And I think
safety, we could have gotten a better sense for that if we had more information about the dose. How many parsley leaves would I have to eat to get, what's the likelihood that a small amount of parsley would have the minimum infectious dose?
Yeah, and, I think you have to extrapolate back from their 25 grams. And that's the only quibble I had with the paper, in that the way they did the experiment is they processed the leaves through the stomacher, and then they did an amplification step, because the diluents that they were using, especially for salmonella, had peptone in. And salmonella will grow quite well in dilute peptone, and it amplifies because they incubated it at 37 degrees for a period, if I'm remembering correctly, about 18 hours. And that's generally enough to amplify it up. And so I don't know whether or not that is an enrichment. And unfortunately, I was traveling.
Well, they use enrichment medium like tetracyanate and novobiocin, which is specific more or less for salmonella. So you enrich for it. And that doesn't tell you what the original amount was.
No, it doesn't tell you if there was one salmonella one salmonella individual on a parsley leaf. So, that's where it's a little bit lacking. But to their defense, they were following the International Organization standardization protocols that were specifically adopted by the European community for the assessment of food safety. And unfortunately, I apologize to the listeners, I didn't have time to go in and compare and contrast to see how that contrasts to what the FDA does in the United States to ensure that our food supply is safe. But the old adage that I learned growing up is you always want to eat local rather than global. You effectively want to know where your food is coming from, and I think we're seeing a lot of that trend by virtue of the fact that we're having farmers markets in our respective communities in which the produce is being grown locally, and it doesn't travel literally farther than the inhabitants who are consuming the produce have ever traveled in their lives.
So, Michael, would washing these leafy vegetables make them safer to eat? Or I should say, how effective would washing be to remove the bacteria?
They really didn't get into that. And you and I were at that session last year with the speaker from the USDA, where he argued that when he eats prepackaged, ready-to-eat lettuce, he still washes it before consuming.
Yeah.
So I think the answer to that question, we discussed that last year as one of the highlights of the GM that you and I actually had attended. I think most people actually wash fresh vegetables before they actually consume them.
Mm-hmm.
So,
well, that's cool. Thank you, Michael.
Well, thank you.
And
so wash your vegetables.
Wash them, exactly.
The take-home message is wash your vegetables, and I guess that's the only other thing is most people actually add the herbs after the food has cooked in order to brighten everything up. I mean, anybody who's ever watched a cooking show know they always add parsley at the end to brighten it, or add the fresh basil and whatnot. What did your mother do, Vincent? I mean, she must have used a lot of fresh herbs.
Yeah, but
her cooking.
She always put it in, and it was cooked. Basil, parsley. It was always cooked. The old Italians weren't much for garnishing.
No, they boiled it to death.
Yeah, and it's got to look good.
Well, they had 7 children to feed, right?
No, we only had 2, but I mean, the family I grew up in, they just cooked it, and it had to look good like that. No garnishing or anything. So we never had any problems, as far as I remember. I certainly have traveled and gotten sick, but not at home.
Yeah.
All right. Well, speaking of that review of last year's meeting, you guys should take notes, and maybe when you come back, you can review what you think are the highlights of the meeting on TWiM.
Okay.
Would that be too much? Would that be too much to ask?
No,
yeah, but we'll do it.
Fun.
It is too much to ask. Okay.
Double our pay.
Oh, well,
double our pay.
Sure, no problem.
Yeah. We'll drag our resident sound mixer Ray along with us, and he can record on the fly.
Yeah, that would be fun. All right, we have a paper for you today, which I have picked because it allows me to talk about translation, protein synthesis, which is one of my interests. And it is a paper in Nature, Structural and Molecular Biology. Title is Proline-Rich Antimicrobial Peptide ONC112 Inhibits Translation by Blocking and Destabilizing the Initiation Complex. And the first author is A. Carolyn Seafeldt, and the last author is C. Axel. And these individuals are from France, the University of Bordeaux, Munich, Germany, University of Munich, and those are the main places here. So this is an international product.
If I could interrupt, there are actually 3 first authors, which you have to look closely at the superscripts to see. But so Fabian Guyen and Stephanie Antunes also contributed, and I'll tell you more about them later.
So where do we see that they're all first authors? Do you have to go to the end of the paper or something like that? I believe you.
It's generally in the footnote.
6, these authors contributed equally to the work.
Cool.
So this
footnote 6, got it. Yeah, I missed that.
Well, it's under author contribution.
Yeah, you're right. First page there. Contributed equally. Wow. 3 first authors. Very good. Anyway, it's a lovely paper. And this has to do with antimicrobial peptides, which are pretty neat things made by multicellular eukaryotes as part of the innate immune response. You get an infection of some sort, virus or bacteria. The cells can make these peptides. They are generally about 12 to 50 amino acids long, and they can inhibit gram-positives, gram-negatives, and even viruses. And there's a category or class of these antimicrobial peptides called defensins, and these are typically found in the fluids covering your epithelial, your respiratory, or gut mucosa, for example. And they can inhibit virus attachment. They can inhibit virus-generated signaling and so forth. And a very famous one is meganin, which everyone must know. And this was, yeah, you should know that, right?
Of course.
It's the very first one because it was,
no idea.
It was found in.
Really?
You don't. He's kidding. He's kidding.
Oh, I should hope so.
Meganin. It was discovered in Xenopus, the African clawed toad, because someone was doing surgery on these frogs, and they would put them back in the dirty tanks, with the womb sutured. And he said, why don't they ever get infections? Tanks are filthy. And it turns out in the skin are these antimicrobial peptides that protect it. And so they're present in many, many different species. And there, as this paper says, there are many that disrupt the bacterial cell wall. And some of them are actually taken into the bacterial cytoplasm by transporters, and they interrupt different aspects of metabolism inside the bacteria. And mammalian cells typically don't have the transporters, so these don't have effects on mammalian cells in general, so they're considered to be non-toxic. People are thinking about making mimics of them as antimicrobial therapies. They can also, some of them can get into the brain as well from the circulation, so they have some interesting uses. So this paper is about an antimicrobial peptide that inhibits protein synthesis, and this has been known before, although the specific mechanism wasn't known, and that's what they go into in this paper. And the peptide they use is called ONC112, which is 19 amino acids long. It's produced by the milkweed bug, which is Onchopeltis fasciatus, and that's where onc comes from.
Hey, can I interrupt?
Yeah.
Tell you, I looked it up, and this is a fascinating bug.
Yeah.
It's a real bug. We call everything bug, but this is a real bug.
True bug.
With legs.
And it eats, it feeds on milkweed, and milkweed is full of toxins. And this guy sequesters the toxin somewhere in the body. So reading up on this is a bug that's used in high school labs and so forth. It's rather common. It's easy to work with. But the website I saw said, please don't eat it because you're going to get very sick from the toxins which are sequestered in part of the body of the bug. That was good advice.
It's a nice looking bug. It's like black with orange highlights, right?
It is, yeah.
So if I could continue this segue, our sidelight, Fabian, one of our first authors, was sitting outside on his campus taking a break from his research, and he saw a red and orange and black bug scurrying around, and he thought, aha, the very bug that we're studying is right here on my campus.
That's funny.
Instead of synthesizing this peptide, I could just catch these bugs. But he took a picture and then went back and realized that it's actually the lookalike bug called Pohacarus Apteris. And if you look it up on Google Images, you'll see that the coloring is very similar, but the patterning is different enough that he went back to his synthesis.
So it must
good thing
be,
he didn't tear it apart and try to isolate it and have the wrong stuff.
It must be that whoever eats this bug doesn't eat the Onchopeltis because it's toxic, and the mimic just takes advantage of that, right? I would guess.
Probably.
Like at the butterfly, the viceroy monarch story. All right, so ONC112 inhibits gram-negative bacteria, and so they want to know how it works. So the first thing they do is to determine the structure, the high-resolution structure of the peptide in the ribosome. So they take crystals of 70S ribosomes from Bacterium Thermus thermophilus, and this is actually one of the keys to getting the crystal structure of the 70S ribosome was to use this species, this thermophilic bacterium. So that structure had been solved some time ago. So they take crystals, they soak.
Is there a reason why they use a thermophilic bacteria ribosome? Or?
It worked, and actually, there's a trio of people who won the Nobel Prize for this, and the lady from Israel, she's the one who figured out that you need to use the high. I don't know why. I think it just worked. She tried it and she got crystals and proceeded, but that's all I know. So they soak the peptide into the crystals of the ribosomes, and they also have, along with the peptide, they add the methionine tRNA initiator tRNA. So this is a special tRNA that has methionine on it, which is going to be recognizing the initiator, the AUG codon. Okay, so this will sit in the P site of the ribosome because that's where normally the initiation occurs, and they also have a little RNA with an AUG codon to make sure that initiator is in the right place. Okay, so to remind you all, not you guys here, but our listeners, when a ribosome attaches to an mRNA in bacteria, there's a Schindel-Garno sequence that helps recruit the ribosome. And in bacteria, of course, the subunits are 50S and 30S. When the two of them are together at the AUG codon, then you have the methionine tRNA, the initiator tRNA, sitting down in the P site. The P stands for peptidyl because that's where the peptide bond forms between the amino acids. And then new amino acids via the tRNAs are added in the A site, and that starts to make your protein. So they put this peptide in to the crystals, and they solve the structure, and they can see there's a beautiful figure showing this peptide is sitting in the exit tunnel of the ribosome. Now, when proteins are made on the ribosome, they come out via a tunnel, which is in the 30S subunit, and this peptide is sitting in this tunnel.
To my untrained eye, this is an amazing fit.
Isn't it beautiful? Yeah.
My goodness, this is incredible.
So it's fitting in there. They can see a little bit of it. They don't see the whole peptide, just a number of residues, but you can see how they're making interactions mainly with RNA, because much of the ribosome, of course, is made up of RNA. There's a little bit of a protein in there, almost as an afterthought, because the RNA is catalytic. But it's making very nice interactions of all sorts with the RNA. Now, the cool thing is So normally the exit tunnel, the N-terminus of the growing polypeptide comes out of the exit tunnel and comes out of the top of the ribosome. But the peptide is inserted in the opposite direction. The N-terminus is down towards the peptidyl transfer site, and the C-terminus is coming out of the outside of the ribosome. So it's a little interesting orientation. So if they compare the structure of this complex, the peptide with the ribosome, with a ribosome structure with only the methionine initiator tRNA bind, they can see that the presence of the peptide induces conformational changes in a number of the ribosomal RNA bases. Okay, so these are bases that are moved around by the presence of the peptide. And these bases, these RNA bases, are also known to move during normal protein synthesis. And during the process of adding tRNAs, charged tRNAs, that means a tRNA with an amino acid on it, there's a lot of conformational changing occurring, and these are the same bases that are changing when the peptide's going, and they call this induced fit. They think the peptide has the right sequence to make the ribosome like it better, to bind more tightly to it.
That explains it a little bit. It explains why the fit is so wonderful.
Yeah, I think that's
a high affinity interaction.
It's a high affinity, right?
It stabilizes that, yeah.
So they can identify a number of interactions between individual amino acids in the peptides and specific bases. In this case, it's the 23S ribosomal RNA. And they can change the peptide, right? They change individual amino acids and then look at the antimicrobial activity. So they can add this peptide to bacteria and figure out a 50% minimal infectivity concentration. And so some of these bases have to be exactly as they are in the peptide. They can't change them. And again, these are ones that are interacting with the ribosomal RNA, and others don't matter at all. So in general, this binding site where this peptide is very conserved, and they say this is consistent with the fact that it has a pretty broad antimicrobial activity among gram-negative bacterium. So the peptide's fitting in this exit channel, making very specific contacts and inducing the ribosomal RNA to fit it even better.
By the way, I was pleased to see a bunch of sucrose gradients.
You bet. They're coming up, yes.
Jeez, that's something I haven't seen for a while.
People still do sucrose gradients, yes. So
do you think this paper was lost in the editor's queue from 50 years ago? Or
Right. No, I think there are. Actually, I don't know if all of this could have been done 50 years ago, but a lot of the technology is
it's new.
It's been around. I mean, the sucrose gradients, the toe printing, which we'll talk about.
But the approaches, though classic, are just so elegant to take the structure and then make mutations to test your predictions and interpretations. It's really beautiful.
Yeah, it's all, of course, having the structure which allows you to guide your mutagenesis. It's really important.
And do functional tests.
Yeah. So now that we're going to talk about the function. What is having this peptide in the ribosome doing? So one way they address this is do in vitro protein synthesis. So they take mRNA and they have an in vitro extract, and it will contains ribosomes and other proteins that you need for initiation and elongation, and they can see protein synthesis work. And they use a technique called toe printing. In this technique, you take your mRNA and you mix it with the ribosome and your initiator tRNA, and then you make a primer, a DNA primer that will hybridize to this mRNA somewhere downstream of the ribosome. And you can add reverse transcriptase. This primer will extend and make a DNA until it hits the leading edge of the ribosome, if you will. Okay, so imagine the ribosome is sitting on the mRNA. You have your DNA primer extended with reverse transcriptase, and wherever the ribosome is, the RT will stop, and then you can run that product on a gel. And if you run it on a sequencing gel next to a sequencing reaction, you can know exactly where the ribosome is sitting on the mRNA. So this is called toe printing because you think about it, footprinting would tell you both sides of where the ribosome is, but this only shows you the leading edge, which would be a toe, right? It's kind of cute.
Cute.
And so they know exactly where the ribosome is. Okay, so they have a lovely figure in the paper that actually illustrates this. Just a little bit more about translation before we get into the results. So after that initiator methionine sits down on the AUG, the ribosome is bound in that region. The next amino acyl tRNA is brought in together with a protein called EFTU. elongation factor TU. And if it's the right tRNA to match the codon that's on the mRNA, there will be a conformational change in EFTU accompanied by hydrolysis of a molecule of GTP. The EFTU will fall off of the tRNA, and then the tRNA undergoes a conformational change to position what's called the acceptor stem, that's where the amino acid is, so that it could form a peptide bond with whatever tRNA is already in there, amino acid linked tRNA. So this whole thing is called accommodation. This probably brings back memories of biochemistry for all of you, but this is an important part of the proofreading step. If the wrong tRNA goes into the site, these changes don't occur and it's just rejected, and that's one of the reasons why we make good proteins most of the time. Okay, so back to their toe printing. In vitro, they can see their ribosomes initiating at the AUG and moving through the open reading frame. They've done a clever thing. They've left out the amino acid isoleucine from the in vitro translation mix. So the ribosome stops at a few codons downstream of the AUG, and they can see by their toe printing assay exactly where this is. Okay. If they add isoleucine, the ribosomes will just run off. So they want to be able to freeze it somewhere. They can also do another type of experiment where they add antibiotics that would prevent the delivery of the tRNA or accommodation. And these get frozen at the AUG codon. Again, you can tell that by the toe printing. Okay, so those are sort of controls for this toe printing assay. Next, they add the ONC112 peptide. They do their toe printing. The ribosomes are stuck at the start codon. They never get beyond it. So basically, in the presence of this peptide, you can get the initiation complex forms. That means the 50S, the 30S subunits of the ribosome are going to sit on the AUG. The initiator tRNA is going to be there, but that's as far as it gets. It doesn't allow addition of a second amino acyl tRNA. So in other words, it's preventing the accommodation step. So they actually predicted this from the structure, which is really cool because the way ONC112 binds to the ribosome would sterically block the 3' end of the tRNA from moving into the ribosomal A site. So they could see there'd be a clash between the two, and that's borne out by this experimental finding. They never get that first amino acyl tRNA in after the initiator because probably the tRNA never gets to accommodate because the peptide is blocking it essentially. And they can actually see in their structure that the peptide is blocking the interaction of a ribosomal and a tRNA bases that would normally base pair during this accommodation process.
It's exquisite detail that they've been able to
It's beautiful.
identify.
It's really beautiful. And, I like this because I've always been interested in translation, but it's just gorgeous that this peptide has this very specific effect. They also notice when they're doing these toe prints that whenever they have the peptide, even though the peptide allows formation of the initiator complex, the 70S ribosome with the met tRNAi at the AUG, it was always weaker than in their controls. So they wondered if there was some clash even between the met tRNA and the peptide, which again was suggested by the structure. So to figure out if that was the case, they made what's called a disystronic mRNA. So they have a single mRNA with two coding regions. Each of them has a Schindel-Garno, which would attract the ribosome, and an AUG codon, and there are two of them in tandem. So in theory, two ribosomes could bind to this disystronic mRNA. And the way the experiment is done, they put this RNA in vitro in an in vitro translation extract, and then they take the product and run a sucrose gradient. Now, a sucrose gradient is wonderful for translation because it can separate mRNAs that have 1 or 2 or 3 or 4 or more ribosomes bound. It's been classically used to study what we call polysomes, which are mRNAs with multiple ribosomes, because the resolution is great. You can separate mRNAs which differ with one ribosome on them, which you can't really do by any other technique as far as I know. So they do this experiment, they run their sucrose gradient, and the first control is to do the translation in the presence of the antibiotic erythromycin, which stalls translation. So the ribosomes will bind and start to move, and then they'll stall. And in fact, what the sucrose gradient tells them is that there are 2 ribosomes on these mRNAs. They see a disome, okay, an mRNA with 2 ribosomes on it. When they have no erythromycin, they don't see anything because the short open reading frames on this mRNA are so quickly translated that the ribosomes run off and you can't see any disomes. They use another antibiotic called thiotrepton, which allows for 70S formation, no elongation. Again, you see disomes. Okay, so these are all really controls to show you that this approach is working. Then they add ONC112 peptide and run the sucrose gradient. Small amounts of disomes are formed. So in other words, they previously showed that the peptide allows formation of the initiation complex, the ribosome on the AUG, but there's much less than you would find, for example, if you freeze the ribosomes with an antibiotic. So somehow the ONC112 is making the complexes unstable. And again, they think this is because where the ONC112 peptide sits in the ribosome, it's encroaching into where the MET tRNAi is interacting with the ribosome, and that causes it to dissociate.
So using this disomic template basically increase their sensitivity, correct?
Yes. Essentially, they can count the number of ribosomes on the mRNA and also see, because I think the toe printing is not really quantitative. It just tells you how far the ribosome has gone, right? But it doesn't tell you how many are bound. But this can do that, right? Now, in the crystal structure that they saw, they didn't see the C terminus of the peptide, which is amino acids 13 through 19. So they do some experiments where they take away amino acids from the C terminus and ask, do these peptides still inhibit? And they can take 7 or 9 amino acids away from the C terminus, and the peptides still will inhibit in vitro translation. Okay? So they ask, what is this peptide, part of the peptide, doing in bacteria? So as I said before, these peptides are generally taken into bacteria by transporters. And this particular peptide, ONC112, is taken by the inner membrane protein called SBMA. And if you make an E. coli lacking the SBMA gene, they are totally resistant to the peptide. So it's beautiful, right? Peptide can't get in, can't inhibit protein synthesis. So this peptide, if they make a peptide lacking 7 or 9 amino acids, it doesn't inhibit E. coli, but it does inhibit translation in vitro. So they think these amino acids are involved with interaction with the transporter protein. So the idea would be the peptide is transported into the cytoplasm by SBMA. The C terminus is important for that. It binds to the ribosome exit tunnel. The ribosomes can make 70S complexes. That is, they can bind to the mRNA. The initiator MET tRNA can sit there, and then elongation is inhibited because the accommodation of the next tRNA amino acid is inhibited. So somehow the C terminus is involved in the uptake, probably interacting with the transporter or something like that. So that's basically what they found. A couple of other points that are interesting. The binding site for this peptide actually overlaps binding sites for other antibiotics that are known to bind to the large ribosomal subunit. So they feel that these antimicrobial peptides can be a new way of looking for new inhibitors of translation that could be new antibiotics.
And something Fabian pointed out is that most of our antibiotics come from bacteria or from fungi, but this is from the insect world. And so he's excited about the potential to identify new antibiotics by shifting to real bugs with legs.
Real bugs with legs. That's great.
So, Michele, did you go to the next question with Fabian and ask him what's the selective advantage of an insect having an antibiotic?
I didn't.
Yeah, that's a good question. I guess when they eat,
they're living in the dirt, right?
Yeah.
So
But protein synthesis is so universal, and you presumably, if insects they need to process their food, having just dealt with termites, I'm well aware of how insects can eat a house, literally. and learned a bunch about that. But it's interesting, unless they're
That's a good
carrion
question, yeah.
feeding where they're eating proteins, and they wouldn't necessarily need protein synthesis per se.
So you're worried that the antimicrobial peptides will harm the symbiotic bacteria in these insects?
Well, in us.
Well, the peptides won't. I see, in us as well. Yeah, that's a good question. Yeah, you don't. I mean, that's a problem with antibiotics, right?
Well, it is a problem with antibiotics, and I think it's a discussion that we're going to begin to see evolve as we begin to understand the importance of the microbiome.
Although they point out here these peptides need specific transporters to get access to the ribosome,
Right.
and that mammalian cells in general don't have the specific transporters.
Right. We're worried about our microbiome, though, being inhibited by
Oh,
this
absolutely.
peptide, right? Yeah, that's an issue. I wondered about resistance because I looked in the literature, and you can get resistance to these peptides, as you might imagine, right?
Sure, just change the uptake mechanism.
You could change that. You could change the ribosome RNAs as well. They didn't really talk about that, but I think that would be a concern as well. And while I was looking at this, I was doing some searches, and I found that another group has recently published basically the same stuff. This is from Tom Steitz's group at Yale, who was he was one of the three who got the Nobel Prize for crystallizing the ribosome, and their paper is in Nature Structural Molecular Biology, same journal. The mechanism of inhibition of protein synthesis by the proline-rich peptide oncosin, which is another word for ONC1112. So,
and is it going to be published in the same issue?
I don't know. It's already out. I don't know if it's the same issue or not.
Interesting.
Yeah. But anyway, it's just an equal opportunity notification. So the paper we just did was published online on
May 18th.
May 18th. And the Tom Steitz one was published on May 18th. Yeah, so it's probably going to be in the same issue, which is good. All right. Yeah, and the last thing I wanted to mention was they do mention this idea. Some of these peptides can get from the bloodstream into the brain. So for brain infections, they could be really useful as well. Anyway, I thought it was really a nice story.
It is, but...
And I love translation, so you can't beat the combination. Yes, Elio.
Could you comment a little bit about how come antimicrobial peptides are found everywhere, including in mammals? We have them.
Right.
Why haven't they broken through the therapeutic barrier and become useful in treatment? I mean, I understand the uptake question is important, but they do get in to bacteria. So how come it's so hard? You read a lot about them. They're found in fungi, they're found in bacteria, they're found in mammals. Now they're found in insects, frogs, etc. What's the story? Why don't we get them at the drugstore?
I don't know the answer, but it may be that, it's a peptide. It's 15 to 20, 30 amino acids, and I think it's therapeutically difficult to deliver and have a good half-life, and so forth, because it's been a while. People have been talking about this for a long time. Michael, you have any insight into that?
I think you hit the nail on the head. It's principally delivery and getting them to the microbes, and they get turned over so quickly.
So cost.
Yeah, I wonder if anybody's done an experiment where they up the dosage of endogenously made peptides in a mouse by genetic engineering, and whether that induces more resistance to bacterial infections or something like that.
I guess the only peptide antibiotic that's actually made it into the mainstream that really doesn't fit the definition of the classic meganin is daptomycin, which is a peptide.
That's right.
How many amino acids is that? Do you know?
I don't know off the top of my head.
It's
I should.
Daptomycin.
Someone can ask Siri.
Yes, ask Siri. How big is But I know daptomycin interferes with the membrane and does. My friends at Cepheid and now /Merck will probably take me to task for not remembering.
Looks like 13 or 14 amino acids, so it's quite big. It's in the range of these AMPs for sure.
But remember, the story of daptomycin is Eli Lilly developed that a long time ago, and it sat on the shelf. And, of course, daptomycin was the founding antibiotic of Cubis.
Cubis, right.
which has just been recently bought by Merck. So maybe, we can ask Barry Eisenstein, an old friend of Elio's, to speculate on why meganins have failed.
So daptomycin inserts into the cell membrane.
Mm-hmm. And pokes holes.
Yeah. It's called cubicin.
Yep.
So today's paper was a little bit different than what we usually do. There was lots more biochemistry.
Yeah.
I'm a little bit concerned about what advice you would have for listeners who are not that versed in biochemistry and they wanted to be able to digest some of this. Where could they go?
I found the paper very accessible.
I did.
They do include growth curves. So
and Sucrose
wild
Gradients.
type and mutant bacteria. So there is that.
There is now the paper may not be open access, but I found a wonderful website. It's at a university. I'll put the link to it, which goes through the steps of bacterial protein synthesis and explains a lot of the. And I think that will be useful because it has actually some movies and some diagrams that are really nice. Yeah. Yeah, I know.
I especially like Figure 5b, where they lay out the target on the ribosome and they overlay each of the antibiotics that's known to bind.
Yeah, it's very nice.
Yeah.
It's really convincing.
The images are beautiful.
Well, I think maybe the message is don't be scared because the paper is actually more accessible than you may think as long as you can speak the language, the basic language of protein synthesis.
I know it's more biochemistry than we usually do, but I think that's good, and I think people like to learn different things, right?
And there's a big review in microbiology and molecular biology reviews that came out in 2005 that's called Inhibition of Protein Synthesis in Bacteria. That's in the public domain, so you can have access to it because it was published in 2005 and it's an ASM journal. And ASM puts out its after the 1-year embargo, you can actually download.
What's the journal again, Michael?
Microbiology and molecular biology reviews, and I'll get the DOI and put it in the show notes.
That's for ASM's review journal.
Right. Well, now we have a couple of review journals. There's the clinical microbiology review journal as well.
So did you learn anything else, Michele?
Yeah, let me tell you about this group. So the first author, Carolyn Seafeldt, was the crystallographer. She had only joined the Innis lab 7 months ago. As a PhD student and solved the structure. And she said it was just terrifically exciting when for the first time she saw the peptide bound in the ribosomal tunnel. So she's obviously off to a great start. She had studied chemistry and biochemistry in Munich at the Ludwig Maximilian University prior to that, earned a master's focusing on biochemistry and structural biochemistry before deciding to move to Bordeaux to pursue her thesis. And that lab, the Innis lab, is especially interested in the 70S ribosome complex and trying to understand the basic steps of translation and antibiotics that can arrest it. She was helped by another collaborator, Bordeaux, Stephanie Antunes, who is a third-year PhD student with Gil Guichard. Her hometown is actually Bordeaux, but she moved to Montpellier in the south of France to study engineering and then worked around the world, including for 2 years in the U.S. She was an intern at GlaxoSmithKline, and then she was a research chemist at a company in Delaware called Air Liquide. And then worked in Geneva at a different company called Firmenich to apply her skills to study and develop compounds that add flavor and fragrance. But then she decided she wanted to get a PhD, so she moved back to Bordeaux, her hometown, and is now studying with Gil Guichard. And their institute is called the Institute of Chemistry and Biology of Membranes and Nano-Objects.
Oh, my.
And then they were helped by Fabian Nagayan, who is in Dan Wilson's lab. And that lab is especially interested in antibiotics and inhibition of translation. And he has an interesting path. He was always interested, he said, in science and technology. But when he was in school, he decided to take what is called real school in Germany, which is comparable, he says, to the British secondary school system. So he got a certificate and then worked in industry, in chemical industry for a number of years, but then decided he wanted to get more schooling and go to university. So he needed to go back to school and do the A-level schooling. He then went to the Ludwig Maximilian Institute in Munich and studied chemistry and biochemistry, and then decided medical research is where he wanted to pursue a career, so he joined Dan Wilson's lab for his PhD. So it was a really nice collaboration between the people who could do the functional tests and the biochemistry, the lab who could synthesize the peptides, including the mutant peptides, and then the crystallographers.
Nice. Very nice.
Yeah. Nice of you to find all this out, Michele.
It was a pleasure.
Did you talk to all these people?
We didn't talk because the time change was a little bit of a challenge, the time difference there, of course, in Europe. But they were quite excited to hear that their work would be featured.
Great. All right. It's a lovely piece of work. There's nothing like a great result to get your career started, right?
Oh, yeah.
that poor girl, the first 9 months, she gets a Nature type paper.
It's tough, isn't it tough? Yeah.
She's going to be very disappointed if
her
luck.
They did point out that they were working some pretty long hours, working pretty intently to get this together. And perhaps they knew that they were under some pressure from a competing group.
Yeah, right.
Oh, I'm sure.
But of course, if they didn't have the crystal structure of the ribosome, it would be a lot harder to do this.
Absolutely.
So that really helps a lot. As they say, you stand on the shoulders of giants, right?
Yeah. You do.
All right.
Indeed.
We have just a couple of email here. The first is from David. Greetings, podcast team. I'm a longtime listener to TWiM and TWiV, and if I had a longer commute, I would get TWIP in the mix too. Only so many hours in the week. I'm a research tech in Portland, Oregon, where our dry, mild winter has given way to a gorgeous, dry, mild spring. Currently 57 F and sunny 2015 to date has been bad for farmers and skiers, but pleasant otherwise. I want to commend you all for taking on papers and questions outside of your specific areas of expertise, because the conversation that ensues lets us listen in on how you think about science questions. If you had all the answers at your fingertips, it would be highly accurate, but of less interest and utility for me, at least. I wanted to weigh in on the mandatory hand-washing sign question because I think it is a Trojan horse. Libertarian politicians and letter writers claim they want to see freedom of choice for consumers, so free markets can make everyone's lives better. But the hand-washing argument is a pretty clear tell regarding their true intentions. Since hand-washing is the slightest of inconveniences to the restaurant worker and has no downside for the consumer, it's hard to believe that they are truly driven by their desire to be able to buy a meal at a restaurant where the employees have not washed their hands. I am convinced, until I see some persuasive arguments to the contrary, that each time you hear about this free market hand-washing topic, the real motivation of the person putting his idea out in the culture is to normalize the idea that government in general and regulation in particular is bad, and they really care about their favorite business interest believed to be oppressed by Washington, not oppressed cooks who'd rather have dirty hands. What I'm really curious is how the independent certification would work. Why are third-party monitors watching us wash up better than a government-mandated sign? If the government was keeping an eye on us in the bathroom, they'd be marching in the streets.
It's true.
So I like this part about taking on things outside of our areas because it lets us learn how you think about science questions. And that's really, I think a number of other listeners have mentioned that. They like to see us deal with things that aren't in our expertise. That's cool.
In other words, talk about something we don't know anything about.
That's right. And eventually get to the right place, right?
Well, look at the discussion we just had about peptide antibiotics, and we tumbled into a discussion on daptomycin.
Yeah, I mean, that's what it is. It's a conversation, and in science, they can be a lot of fun. Right. The other one is from Bob. Hello again. I just listened to the Red Queen's discussion about iron battles on TWiM 103. Regarding your interest in other occurrences of this phenomenon, there is a paper by Roy Jensen et al., where he discusses in detail the battle for tryptophan between chlamydia and their mammalian hosts.
Oh.
So there are other battles out there.
That's interesting. Tryptophan.
And remember, chlamydia is obligate intracellular parasite.
Right.
This is interesting.
Look into it.
Yeah, look at this paper. It's full of nice diagrams. Frontiers in Cell Infection and Microbiology. Interesting journal. Open access. Everybody can read it. Well, thank you, Bob. We only have 2 email. So, folks, start firing up your word processor.
And remember that the election is not until 2016, so we can go easy on the political.
Can I make a little statement about occasionally emails which are meant for This Week in Microbiology make their way first to Small Things Considered. Well, I just send them over, so you can avoid that intermediate step by sending them directly to TWiM. We're not equipped to handle the same questions in Small Things Considered the same way.
Yeah. Yes, do send them directly.
But it doesn't happen often, but it happens once in a while.
I think sometimes you send them to me.
I always send them to you.
Put them here, yeah. This hand washing thing has got some topics really get people's interest. This has been one we've been hearing about for a couple of weeks now. It's really curious to see what people get excited about,
I'd put vaccines in the same category. To many of us, it seems black and white, but not to everyone.
Seems to me
I don't know where you draw the line. I mean, you cannot be absolute about it. Otherwise, there would be no traffic lights.
That's right.
That's right.
You couldn't drive, leave alone get a driver's license. So somewhere you have to compromise. There is no such thing as absolute freedom. You couldn't live in a civilized society without some compromise. So where do you draw the line is probably an individual question. But you have to draw the line is the point.
Yeah, for sure. All right, TWiM 105. You can find this at iTunes and at microbeworld.org/TWiM. There's also a Microbe World app you can use. There are iPhone and Android apps that you can use to download podcasts. You'll find TWiM on all of those. And we'd love to get your questions and comments. Please send them to twim@twiv.tv, not the Small Things Considered. twim@twiv.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you. See you next time.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
My pleasure.
And Michael Schmidt is usually at the Medical University of South Carolina. Thank you for joining us from New Orleans, Michael.
Well, thank you. You'll be missed this week.
I don't think so, but you'll have a good time. You don't have anything to do today, do you, Michael?
No. I came in because I have an 8 o'clock meeting tomorrow morning, and when you fly, it's in thunderstorm season. It's best to do it in the morning.
Oh yeah. Well, you can go walk around now. Have fun.
Yeah.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and in particular Chris Condian and Ray Ortega for their technical help. The music you hear at the beginning and end of TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.